Okay, so hello everybody for the usual Friday recorded class, and I hope you all had a good Thanksgiving. Please do take the time to watch this video carefully before we proceed next week. For all of the rest of the semester, we're going to be discussing bacterial diseases. There's a huge amount that we could talk about. We have to just skim through just to give you an idea of some of the variety and some of the methods by which bacteria end up causing disease. So that's the first thing. Second thing, I just want to remind you about this construction of the body, which creates areas of the body which are richly invested with a normal natural microflora. That would be our skin, the interior of the mouth and the nose, then the gut structure as well. But what you need to think about is that all of those microbiota organisms exist actually outside the body. Even those which are in our gut are actually outside. And the gut lumen, all the way from mouth to anus, that is essentially a tube. And the interior of that tube is outside the body. And bacteria are prevented as far as possible. They are prevented from crossing barriers into the interior of the body. I call it the sacred interior, because the interior of the body, the gut cavity, the thoracic cavity, the brain, these are kept bacterial free as far as possible. And in order to get bacteria to pass into the sacred interior, those barriers need to be breached. Sometimes those barriers are breached as a result of wounding. That is one way in which bacteria can make their way into the bloodstream, for example, or into the interior, in the muscles or whatever. The other way that happens is that certain bacteria may actually possess pathogenic qualities which enable them to breach the barriers. They possess things which, for example, may dissolve cells, kill and dissolve cells of the barrier. Now, on the outside of our bodies, we have thick skin, which is salty, and which also contains, our sweat releases antibacterial compounds as well. On the interior of our body, we are coated with a very profound barrier, which are the mucous membranes, but they are thin and they can be relatively easily breached. We're going to hear how those surfaces are actually managed, especially the ones on the interior. We won't talk much about skin diseases and things, but we'll hear a little bit. But most of our important bacterial diseases breach the barriers either of the upper respiratory tract or else of the gut. Those are the two major inroads into that sacred interior to cause serious illness. And each of them has to involve some kind of breaching of the protective barriers. So it's not surprising that in all animal populations, but we're considering human populations, it's not surprising then that the two groups of diseases are bacterial diseases, which are of the greatest importance and have the biggest impact on human mortality, are respiratory diseases and then diseases of the gut, because those are the two major ways that bacteria can gain entry into the sterile interior of the body. So we're going to talk about diseases of the respiratory system first of all. Hello, doesn't want me to go. So I'm not going to go through all of the anatomy in any great detail. I just want to emphasize that our respiratory system is divided into an upper region and a lower region. And the upper region is by no means sterile. it has a rich microflora, a rich microbiota. Thousands of different species inhabit our mouths, our nose, sinuses, and down into the throat. But the lower respiratory tract is by and large kept bacteria-free. It is pretty sterile. Some bacteria, few bacteria may make their way down, but there is a very, very powerful protective mechanism. So each of those, the upper respiratory system and the lower respiratory system, have to deal with the presence of bacteria, but they do it in different ways. The upper respiratory tract, the mucus of the respiratory tract produces substances which in particular are active against gram positives. One of the things that they produce is an enzyme called lysozyme, which breaks down the gram positive cell wall. Remember, whenever I say those words, cell wall, go back either in your mind or in your notes, go back and look at the structure. The gram positives have a thick cell wall, and it is there to provide osmotic protection against bursting. But if it is attacked either by the substance lysozyme, the peptidoglycan breaks down and the cells burst. So there is a specific aim of producing a lysozyme. In the lower respiratory tract, the mechanism is completely different. The lower respiratory tract is coated with ciliated epithelium, cilia which continually beat, and the power stroke is upwards. So anything on those cilia tends to be brought upwards. In addition, the surface of the mucosal surfaces, both upper and lower, produce mucus. And in the lower respiratory tract, it forms a continuous sheet all the way from the small alveoli in the lungs, all the way up to the top of the throat. And that is called the ciliary escalator, because it is a sheet of mucus that is continually moving. And that sheet of mucus traps any bacteria that might find their way into the lungs or any part of the lower respiratory tract. It tends to trap bacteria and rapidly move them up and away from the lungs. They are moved up to the top, to the pharynx, at the top of the throat, where we swallow it. The mucus that you blow out of your nose is not mucus that's come from down here. It's mucus that has been produced up here. Now, a similar idea does pertain in the upper respiratory tract. We have cilia beating, which move mucus down to the lower part of our nose where we blow it out. That is like trapping and removing, trapping, moving the bacteria the whole time. It's a very powerful mechanism. And as we go lower into the lungs, which become very, very vulnerable to any bacterial infection, there are additional protective mechanisms. And in the lower lungs, there are dedicated macrophages. Those are white blood cells which patrol and which not only eat bacteria, but I've just got to do something. I'm sorry, I hit a button. Sorry, I hit a button that I shouldn't have hit. Okay, so in the lower parts of the lungs, in the alveoli and in the small tubes leading to the bronchioles leading to the alveoli, there are dedicated macrophages which will not only eat the bacteria that they encounter, but which will alert the immune system to the fact that that bacteria are present. And in addition, there are substances released in the mucus which tend to retard the growth of bacteria enough so they get trapped in the mucus and conducted up. I labor the point because these are extremely important protective mechanisms. And if they fail, then bacteria can very rapidly grow and populate what should be a sterile space. So I'm not going to go through all of the anatomy. This is the upper respiratory tract. I'm just going to mention the most important things to take note of. First of all, it's divided, of course, into the mouth cavity, which has its own microbiota, but also is subject to infection by pathogenic bacteria. This upper chamber here, these are the nasal passages and the sinus connected to the sinuses. Again, coated with mucous membrane, ciliated epithelium, which moves mucus down to the front of the nose. There is one important feature that I want to point out, and that is this little fine tube, which is the eustachian tube, which connects the pharynx. This is the pharynx here, the common tube, before it divides into the airways and into the esophagus. So this is the pharynx here. And the eustachian tube connects the inner ear, the middle ear with this cavity here. And that's to allow us to equalize for atmospheric pressure between the two, because the middle ear is filled with air. It's lined again with ciliated epithelium, but it needs to have a mechanism to equalize pressure. This is probably familiar to any of you who've taken a plane flight. The last thing I want to point out is this little, it looks fairly obscure, but it's extremely important. This is the epiglottis. Whenever we swallow something from the mouth, through the mouth, the epiglottis slams closed over the trachea. This is the trachea here leading to the lungs, and that is to prevent food making its way into the trachea. When people choke, very often either food has lodged here and the epiglottis has closed off the airways and food is lodged here, or else something has made its way down into the trachea and blocked it. So the reason I point this out to you, people often confuse the epiglottis with the uvula. And the uvula is that little flap you can see when you look in the mirror at the back of your mouth. That is not the epiglottis. The epiglottis is further down at the back, down below the tongue. And in bacterial disease, this is one of the most important sites, because infection and inflammation of the epiglottis is a critically dangerous situation, because if the epiglottis swells, it blocks the airway. It also loses control of the position of the epiglottis, but it swells and can block the airway. This is especially important in children. Okay, the lower respiratory tract. Yeah, the trachea, the big tubes, the bronchi are the tubes leading away from the trachea into the lungs. The very fine tubes leading to the alveoli are the bronchioles, and these are the alveoli here, and they are always filled with air, and that is the action of respiration. Circulates the air, pumps the air out, and brings the air in. But every time it brings the air in, there is a possibility that we could be bringing bacteria in. That's the function of the escalator, the mucus escalator that moves the bacteria out. So depending on what part of the anatomy is being infected or by bacteria, it usually produces an inflammation. A pharyngitis is the typical sore throat that we get. Now, people will often say, I've got strep throat. There are many things, many bacteria with pathogenic qualities that can cause a sore throat. But this is a typical manifestation. It's in the upper part of the throat. Laryngitis arises when the larynx becomes inflamed, and especially when the vocal cords become inflamed. And laryngitis, also probably familiar to most of you, is when we lose our voice simply because the muscles are inflamed and don't work properly. Tonsillitis is an infection, persistent infections of the tonsils. I'll just flip back. The tonsils lie here. Sorry. The tonsils lie here at the back of the throat there, on either side. In the old days, children used to routinely have their tonsils taken out, whether or not they were ill. Nowadays, it's recommended people only have the tonsils out if they have persistent tonsillitis, that is infection of the tonsils. The tonsils are part of the immune system. They're a very important part of the immune system, but we can live without them. But the structure of the tonsils, I can only just show you by using my hand that they have these cavities leading into the interior of the tonsils, which catch bacteria and things and conduct them down to areas of the tonsils where they can be dealt with by the immune system. But certain bacteria, if they infect the tonsils, they evade the protective mechanisms and can start to multiply down in the tonsils themselves and cause tonsillitis. That is really quite common in children, but nowadays they mostly opt for treating with antibiotics rather than immediately removing the tonsils. Sinusitis, this is something probably many people are familiar with. Sinusitis are infections of the sinuses. Sinuses lie, the biggest sinuses lie up here, but there are also sinuses in the bones here. They connect to the nose and normally drain freely into the nose, but they can become infected with pathogenic bacteria which multiply in them and cause inflammation. And very often that leads to blockage of the flow of mucus from the sinuses, and that produces the typical sinus headaches that are characteristic of sinusitis. Epiglottitis, remember, I've described this to you already. Epiglottitis is a very, very dangerous inflammation of the epiglottis, which can lead to airway obstruction. All right, so I've described to you already the different compartments of the respiratory system and the upper respiratory tract with its rich microflora, but the lower respiratory tract with very, very few bacteria. Here's a picture of the mucous membranes of the respiratory tract. And typically, this is ciliated epithelium and interspersed with these large cells which produce the mucus. They produce it continuously so that it forms a sheet. This is an electron micrograph, scanning electron micrograph of these ciliated cells. These are extremely fragile. They are protected. They themselves are protected by the mucus, but they are easily damaged, and certain pathogenic bacteria are actually able to settle on the cilia and cause them to stop beating. And then those bacteria very often will strip the cilia off to gain entrance to the cells of the mucous membrane. So let's talk about some specific disease. The first one is one which automatically sounds familiar to us all, and this is strep throat, streptococcal pharyngitis is the proper term for it. And as I said before, often people say, I've got a terrible sore throat, I have strep throat. Might not be strep, in fact. There are numerous bacteria, pathogenic bacteria, that can cause a strep throat. That soreness arises from two different mechanisms. The first is that the bacteria are actually damaging the cells of the pharynx. The second is that they are exciting an immune response, which causes inflammation and, in addition, causes added pain. One of the features of an inflammatory response in the body is that it increases sensitivity to pain. So that is one of the reasons why sore throats can be really so painful. And of course, we swallow the whole time. Every time you swallow, you have a shot of pain to remind you that you have the infection. But just to emphasize, remember that if you've got a sore throat, you actually have to be tested to ensure that it really is strep throat. So pathogenic, there are numerous members of this genus Streptococcus. And the commonest Streptococcus that causes illness is Streptococcus pyogenes, which is actually a normal common skin bacteria. It often establishes an infection in the throat after a viral infection. Not always, but often the bacterial infection follows on a viral infection, which damages the mucous membranes and allows the bacteria to adopt a pathogenic strategy. But Streptococcus, especially those that belong to this group, they're called Group A Streptococci. Streptococcus pyogenes is a member of the Group A Streptococci, or the GAS. So they have some innate features which allow them, if they're given the opportunity, allow them to be opportunistic pathogens. And the first is that they have a thick capsule around them and a couple of other features which make them resistant to phagocytosis by macrophages and other cells of the immune system, which are normally the first line of immune response. The second is they possess a set of enzymes called the streptokinases. And the streptokinases break down blood clots. Now, I can't go into all of the inflammatory responses, but one of the responses of the body in inflammation response is to cause clotting around an area of infection so that the infection is limited. The ability of this infection to spread is limited. The group A streptococci produce streptokinases which break down those clots so they are able to spread through the tissues and sometimes into the bloodstream. And the soreness of a sore throat caused by strep is because they release streptolysins, and these are the enzymes which break down cells. So they're cytopathic, so they will break down cells and damage them. All of this will cause a profound inflammatory response by the immune system, and very often in a true strep throat, that will be accompanied by fever and by enlarged lymph nodes as well. I'll repeat that to know that you've got strep throat, you actually have to be tested. But you can have that done in a doctor. The doctor can do that in their office. There are rapid tests that they can do to see whether you do indeed have strep throat. It's important if you've that ill that you're needing to see the doctor. It's important to establish exactly what it is so that appropriate antibiotics can be prescribed. But here's something really, really interesting about these streptococci. And that is they are subject to infection by a lysogenic virus. Now remember what the lysogenic life cycle was. The lysogenic cycle in viral infections of bacteria is that the bacteria continues to live and to function. The viral genome becomes integrated into the host's chromosome and is read off periodically as the cell divides, okay, to replicate the virus. And then eventually it will cause lysis of the bacterial cells. But in the meantime, some of those viral genes can actually be read off as if they were host genes. So this, what happens is that they begin to, especially as the approaches the stage where the virus is going to cause lysis, they start to produce a set of enzymes, viral enzymes, called erythrogenic toxins. And those, as the bacteria that are infected with the virus, as they begin to lyse, they release this erythrogenic toxin into the bloodstream. It's not a matter of the bacteria setting up septicemia. Bacteria can be in one location, but they release toxins into the bloodstream, which have a systemic effect. So first of all, this is a typical sore throat. This is just a bacterial sore throat. And just looking at it, you can't tell that it's Streptococcus. But there's whoever took the picture shows us that this really was Streptococcus. The typical inflammatory response, this bright red, swollen sore throat. These at the back here, this is not, they may look like highlights but they're not. These white specks here are bacterial colonies growing on the mucous membranes around, and at the back, here are the tonsils, also infected. But the release of that erythrogenic toxin produces some very distinctive effects. And the first disease that we talk about is this disease, scarlet fever. This is due to Streptococcus pyogenes, and the genes, the viral genes, cause the cells to make, the bacterial cells to make this erythrogenic toxin. When they lyse, they release it. And this is the effect. The first is people often develop this a very distinctive, smooth, inflamed tongue that is called glossitis. And it's also referred to as strawberry tongue because it actually ends up turning this bright strawberry red color. And the rest here, this is where it gets the name erythrogenic. Erythrogenic toxin is one which causes a rash, causes this macular rash all over the skin. And that's why it gets the name scarlet fever. So the scarlet fever it used to be serious, especially in children. It was a cause of mortality in children. But nowadays it's relatively easily treated. But the problem is that you have to do it very judiciously, because you can't kill off that whole bacterial population. They'll release so much erythrogenic toxin that it would be dangerous. So there are some difficulties in managing the disease judiciously. The really dangerous thing about scarlet fever, normally people recover spontaneously. As the bacterial population dies off, then they recover. But there's a really dangerous aspect to this, and that is that the bacteria also contain a protein called M protein, which is very, very similar to endothelial proteins in human beings. And if the immune system reacts against the bacterial M proteins, they can set up an autoimmune disease called rheumatic fever. And it is called rheumatic fever because it produces intense joint pain, but it can also attack the valves. Your own immune system attacks cells of the circulatory system. And this the commonest effect of this was to damage the valves of the heart. So that is the condition rheumatic fever, also a result of the Streptococcus infection. So let's have a look now at another bacterium. This one is used to be spectacularly important in human populations right up until the advent of antibiotics. And this is a bacterium called Corynebacterium diphtheriae. Well, that tells you right there, diphtheria. This is the cause of the disease, diphtheria. It's a rod. It's quite a long rod, and it's unusual in that it's gram positive, but it doesn't form spores. And it's Corynebacterium diphtheria. It is relatively contagious, so it can be commonly was spread from child to child in schools, or just in contact, because it would settle in the lungs, and once it does, it actually settles quite easily on ciliated epithelium, and it produces some very, very distinctive effects. It's the pathogenic qualities of the bacterium lie in the fact that it causes intense inflammation, but the inflammation builds up what is referred to as a pseudomembrane. And the pseudomembrane is a mixture. It's clotted serum from the bloodstream mixed with dead tissue, which is sloughing off from the damaged mucous membranes, all bound together with mucus, which is consolidated. So it forms this thick, tough, grayish, it looks like a membrane. Of course, it's not a cell membrane in that sense, but it is a thick membrane formed as a result of pathogenic genes of Corynebacterium. They also produce an exotoxin, a toxin which is released from the bacterium. And that, again, is due to lysogeny. That gene is imparted to the bacteria by the virus integrating into the genome. And as the cells die, they release it, and that can cause heart and kidney disease as well. So this is a very dangerous bacterium, especially in children, and I'll show you exactly why in a minute. We now vaccinate. We vaccinate against the diphtheria toxoid. The toxoid are those enzymes released by the bacterium, either innately or as a result of lysogeny by a virus. And the vaccine is against that toxin. As long as the ability of that toxin to operate is removed, the bacteria are easily dealt with by the immune system. It is the ability to produce these toxins rather, that gives Corynebacterium its pathogenic quality. So here is typical symptoms of diphtheria in children. It used to be one of the leading causes of infant mortality, and all through history, right up until the advent of antibiotics, especially in the 19th century, it reached a point where most families could count on losing at least one child to diphtheria. It was absolutely dreaded. And the reason for that is this. First of all, this is the result of the exotoxins being released and causing this rash with a very high fever, which itself could be dangerous in infants. But the most dangerous thing of all was this infection of the pharynx, and here you can see there is a pseudomembrane, and you can see that this pseudomembrane has blocked off all of this side here. It could also spread down to the epiglottis. And you can appreciate that if that pseudomembrane formed on this side as well, it could lead to a blockage of the airway, and that's exactly what would happen with children. There were doctors who specialized in coming around and lancing this pseudomembrane and removing it, an intensely painful experience for the child, but it could save their lives because it would open up an airway for them. But many of them died as a result of the circulating toxins, fever and everything else, but then of the blockage of the airway as well. A very terrible way to die. This is now prevented by the DTaP vaccine. top diphtheria, tetanus, and pertussis vaccine. Very good vaccine, and one which is routinely administered now to children. Yeah, the bacteria occur commonly, but if it's only when they possess this virulence factor, which releases the toxin, that they are able to cause disease. Just as an example of the success of the vaccine, in the United States, there's now about 5 cases of diphtheria recorded, often from people who haven't been vaccinated. When it occurs, it's from people who have not been vaccinated or for whom the vaccine didn't work for one or other reason. Okay, so in children, these respiratory diseases often are particularly important for infants and for young children. And that's because of the narrowness of their airways and just of their anatomy in general. Here's one also familiar, and why can't I go backwards? Anyway, just go back to that anatomy slide that I had before, and familiarize yourself again with the eustachian tube. Remember that the eustachian tube connecting to the middle ear is also invested with a mucous membrane. So is the inner ear. They have their own microflora, but the middle ear continuously drains into the back of the throat, and it's to equalize atmospheric pressure, but also to provide an outlet, a continual sweeping out of bacteria that might be breeding. If you block that eustachian tube, even if you, in the absence of a pathogenic bacteria, the growth of bacteria inside the middle ear causes problems. This is exacerbated if a pathogenic bacterium has made its way up the eustachian tube into the middle ear. And in children, this is extremely common. It results in intense inflammation, but also in the production of pus in the middle ear. It can be caused by a wide variety of different organisms. Streptococcus, the Streptococcus pneumoniae, that's a common one, or Streptococcus pyogenes. Another bacterium called Haemophilus influenzae. Haemophilus influenzae has nothing to do with influenza, nothing whatsoever to do with influenza, but it commonly occurs as a result of having had influenza. Another one called Moraxella. There are various others, but it can also be caused, this blockage of the middle ear can also be caused by viral infections as well. And it's a symptom, for example, of respiratory syncytial virus, which is currently enjoying an epidemic in children. And this impact of this is that those of you who go into nursing or medicine, you'll commonly see this picture. When you look into the ear of a child screaming with earache, with intense earache, and you see this. This is the tympanum here, and that's the membrane at the end of the ear canal, which is the boundary between the passages of the ear and then the middle ear on the other side. Remembering the middle ear only has one outlet, and that is through the eustachian tube. Block that and fill the middle ear with bacteria and with pus, and this is the picture you get. The pressure builds up inside so much that it ends up pushing the eardrum out, and it can actually spontaneously rupture in very severe cases. This is normally treated with antibiotics, but if it persists, then the easiest way to treat is to provide another drainage mechanism, and that is by putting grommets into the eardrum. These little plastic makes a little hole lined and stabilized in the eardrum that pus can drain out of. Those grommets, by the way, don't have to be removed. They are spontaneously healed over, and the grommet is expelled Okay, so that's just a couple of the diseases of the upper respiratory tract. Remembering that the most dangerous ones are those which produce exotoxins of one sort or another, or which might make their way across the mucous membranes into circulation. The lower respiratory tract infections are all potentially serious because that is an area which is supposed to remain more or less sterile. Bronchitis is the inflammation of the bronchi themselves, familiar to all of us who've had a terrible racking cough due to bronchitis. Bronchiolitis is much more serious because it is an inflammation of the very small tubes leading to the alveoli. And that bronchiolitis very easily leads to a pneumonia. Pneumonia strictly refers to a symptom, not a disease, but a symptom. And that is the filling of the alveoli with fluid. And the obstruction of the alveoli, which does not allow them to pick up oxygen and release carbon dioxide. We say the lungs become consolidated, they become fluid filled once they've been infected and inflamed. Bronchiolitis very easily leads to pneumonia. Pneumonia can arise simply because of infection of the alveoli as well. So a couple of these diseases you may have heard of. Fortunately, at your age, most of you will not have had these diseases. But at my age, when I was a child, these were extremely common because there were no vaccines against them. The first one is called pertussis, and the DTaP vaccine is against pertussis. So it's diphtheria, tetanus, and pertussis vaccine. It's caused by an organism called Bordetella pertussis. It's a gram-negative, and it produces a capsule which enables it to adhere directly to the cilia of the ciliated epithelium. And because of this, it is able to establish itself directly on the cilia and spread through areas of the trachea. It destroys the ciliated cells with a set of exotoxins, and it's actually a really nasty bacterium. It produces a range of toxins which shut down the ciliary escalator. And as a result, usually a child, the only way to void the lungs is to cough up the mucus that is accumulating because the bronchociliary escalator has failed. And they do so in paroxysmal coughing. The coughing develops so much that they actually almost empty the lungs. Very frequently you'll see the child over in a coughing fit, left at the end with absolutely no air. And when they come up, they go as they pull air in recovery. And that whooping, excuse me, myself whooping cough. That whooping is what gives it its name, whooping cough. But the pertussis toxins also cause systemic effects as well, which can be extremely serious. And in particular, they cause a very high fever. Pertussis is used to be a very dangerous disease in children and a major source of infant mortality. So these are the different stages. It is not that commonly seen anymore because of the success of the DTaP vaccines. It starts like a common cold, and then it progresses to this paroxysmal coughing stage. And it then goes into a convalescent stage. The problem with it is those toxins, even after the bacteria is gone, those toxins are very long-lived, so they can cause very long-term sequelae, especially in children. It can also be treated with erythromycin and earlier antibiotics, but only if they're used early on. Don't forget, If you've got a large bacterial population and you lyse them all, they release all of those toxins are released at once and may cause even greater problems. Okay, so we now proceed to one of the most important of all human diseases. This is sometimes being referred to as the great malady. It's the disease of tuberculosis, and it is still a source of major human morbidity and mortality worldwide. In fact, there's been a resurgence of tuberculosis cases all around the world for reasons that I'll explain. It's been recorded even in fossilized human ancestors' bones. So it's it has been around for many hundreds of thousands of years in the human population. And it's caused by a bacterium, Mycobacterium tuberculosis. And you'll remember, mycobacteria they're actually fundamentally gram-positive, but they have an additional component in the cell wall, which is a waxy substance called mycolic acid. It gets its name from the bacterium. It's called mycolic acid, and its presence means we can't gram stain them. And we actually have to use a special staining method. Go back to your laboratory's notes, because you will have heard about the acid fast test. It's a special staining technique which reveals the presence of mycobacteria. It has a very slow, it's very slow growing, which is a problem in diagnosis. It takes a long time to culture mycobacterium clinically. And it is very resistant because of the waxy coating. It's very resistant to drying and to antimicrobials, which is a problem, especially in households with a member who has tuberculosis, because the bacterium can be long-lived in aerosol. Although the bacterium itself is not very contagious, it's not highly contagious, but it is contagious, especially in intimate circumstances or where you're exposed to aerosol from somebody coughing. One of the symptoms, eventual symptoms of tuberculosis, is that people develop very severe cough, which may release not only mucus, it may release blood droplets as well from the interior of the lungs containing the bacteria. Very high doses of the bacterium will cause disease, but you do need quite a high dose. We have some other related diseases. We can also pick up mycobacterium from cattle, and this is called bovine tuberculosis. And it's about 1% of US cases. That's quite a high proportion. There's another one called Mycobacterium avium intracellulare, or MAI. MAI is a related mycobacterium, but it only occurs usually in birds. And if we ever breathe in, for instance, it can be acquired from bird feces. And it's so resistant to desiccation that the bird feces can dry and blow around as dust, and you can breathe them in. In anyone with a normal functioning immune system, our immune system responds extremely vigorously and wipes it out. But if your immune system collapses, as in HIV, for example, then the disease can establish. And the presence of Mycobacterium avium intracellulare, an almost unheard of infection in human beings, was one of the hallmarks of the start of the AIDS epidemic. And that MAI became one of the killer organisms that would kill people who had AIDS, full-blown AIDS. So here's the bacterium. You can see it's difficult to make. Let me try and find it. Here's an individual one, this long rod here, stained red by the acid-fast reactions. But they often grow together in palisades, that is stacked against one another, or in cords where they all grow together to form these long bodies of cells packed together. So this would be a good example of a sputum sample from somebody who is suffering from tuberculosis. So there's something that explains why it is so important as a human disease. First of all, when you acquire the bacterium, the first thing that happens is that our immune system recognizes the presence of the bacterium very, very easily. There, in fact, are the interaction between this bacterium and our immune system has gone on so long, for so many hundreds of thousands of years, if not millions, that they have developed this dance that they play with one another. There are elements of our immune system which are specifically there to deal with tuberculosis. But likewise, the tuberculosis bacterium has acquired features which enable it to evade those immune responses. So the first thing that happens is that bacterium gets ingested by a macrophage. And the macrophage is going to send out messages to the immune system to alert that something is up, that something is going on. And that will end up summoning other macrophages. The problem is this: the macrophage eats the bacterium supposedly to break it up and to digest it and use it as nutrition. That doesn't happen. The tuberculosis bacterium evades the ability of the macrophage to digest it, and it starts to breed inside the macrophage. More macrophages come, and if that the macrophage will eventually die, and they have a relatively short lifespan, the infected macrophage dies, and more macrophages become infected, all centered around the original area where the bacterium was first ingested. And they begin to form this structure. On the inside, there is an area where the bacteria are freely dividing. On the outside, the immune system responds by building up like a shell around it. And that structure is referred to as a tubercle. And the inside is kind of dead cells, and which the bacteria love, and they feed off the debris and everything from the dead cells. And the other cells of the immune system crowding around now begin to form this protective shell called the tubercle. Now, this is not full-blown disease, but the tuberculosis bacterium is actually quite a toxic bacterium. It releases toxins which go into circulation, and the person may indeed begin to feel symptoms even at this stage with a small center of infection. But I'm stuck. Why am I stuck? Just let me try one thing. I'm sorry, I apologize. No. In any case, I can just describe to you what happens while my computer's evidently sitting and thinking. This tubercle eventually becomes invested with other cells of the respiratory system, and it goes silent effectively. As long as the immune system is able to maintain vigilance over that tubercle, there is a strong possibility that the person will only suffer from a latent infection. The problem comes when the bacteria on the inside are able to break through this protective outer covering. and the bacterium then becomes disseminated. Disseminated very often first in the lungs. So they begin to form tubercles all over the place, but they begin to occupy the alveoli and to spread. One of the impacts is they begin to cause cell death, and lung tissue begins to die off. They're actually opening up holes, literal holes in the lungs. The second thing is that they may be able to gain access to the bloodstream, and the bacterium can actually spread, and it can be located, end up being located in the brain, in bone marrow very frequently, in the organs, and you then have disseminated tuberculosis with the same small tubercles forming all over the body as an initial response, but those tubercles breaking down, so that resulting in tissue damage. And that is referred to very often as miliary tuberculosis or disseminated tuberculosis. On X-ray, that appears as these tubercles appear all over the body. Let me try again. I'm dead. My computer's decided to go night-night, so let us all go night-night. I'll finish this section when I see you on Monday. And until then, enjoy the rest of your weekend, but please do look through this and through the PowerPoints. Let me see. Wait a minute. Wait a minute. Don't go away. Don't go away. There. Now I've just got to get me back. Right. Sorry, we're not finishing. Sit right where you are. Sit. There. Okay, so here is what I was talking about. Here's the mature tubercle, and here's all the bacteria trapped on the inside. Here's an outer layer of immune cells, and then often on the outside there will be a layer of protective cells. But eventually these bacteria are able to break through this and spread around, initially around the lungs, but then around the rest of the body as well. So tuberculosis is a very, very dangerous disease, and it is a very widespread cause of human mortality. There have been various attempts to develop a vaccine, but these really have very little positive impact. There's one called the Bacillus Calmette-Guerin vaccine, or BCG, and that was developed from tuberculosis of cattle, bovine tuberculosis, Mycobacterium bovis. It has some efficacy, especially in children, but the efficacy, it fades, and it has very little impact in adults. They do not develop a strong immunity at all. For those of us who grew up in developing countries, we all had the BCG vaccine. None of you probably would have had the BCG vaccine unless you've come from a country where it is still used. In the United States, it is not usually administered. So this is a progression of tuberculosis. Here's what would happen: the initial infection, the initial tubercles, but they spread out. They'll spread locally, cause tissue damage, and then enter into the bronchioles, into the other alveoli, and eventually start causing the lung function to be severely impacted. And this is how they appear. You can see in the rest of you can see the tubercles spreading through the lungs. You can see holes opening up where the tissue has actually died away. So there are ways to test people. The first way is to do a tuberculin skin test. They just stamp you just under the skin with a small amount of the toxin, of tuberculin toxin. And if your immune system responds, it suggests that your immune system recognizes that. And the only way your immune system could recognize that would be if you had been vaccinated against tuberculosis, or you have been exposed, or you have an active infection. So for a start, this is one reason why in the United States they don't administer the BCG vaccine. I can't have this tuberculin skin test because I had the BCG vaccine, and probably because I'm from Africa, I have probably been exposed to tuberculosis. The actual diagnosis needs to be made by X-ray, computerized tomography, and then also the classic way is to get a sputum sample and test for acid-fast staining bacteria. But now there are also blood tests and serology tests that can be run. Realize that in what is happening in tuberculosis is the bacterium is actually manipulating the immune system. The formation of the tubercles is an immune response, but there comes a point at which the tuberculosis can actually break through and evade and disseminate. So as I said before, it's like a dance between the bacterium and the immune system. So it is treatable, but with one caveat, and that is that tuberculosis, unfortunately, is highly mutable, and it very, very rapidly develops resistance to the antibiotics. The first antibiotic that was of major impact was streptomycin, and that was in the 1940s, and that led to cure of many, many millions of people. But it very rapidly became useless because the bacterium became resistant. So currently, if you suffer from tuberculosis, the first drugs that they will try are these, any one of these, and isoniazid and rifampin are probably the most common. Many tuberculosis strains are resistant to those, however. So then they will try these stronger antibiotics. They will still use streptomycin in conjunction with other antibiotics. But unfortunately, there are two groups of tuberculosis which have become extremely common. And those are the multi-drug resistant tuberculosis strains, which are resistant to all of these. To all of these, sorry. They are resistant to all of the first-line drugs. They will still respond to second-line. And those are the multi-drug resistant tuberculosis strains. And now there are also another group called the extensively drug resistant, which are resistant even to these. And there are numerous new antibiotics being tried to deal with the extensively drug-resistant strains. Tuberculosis is a worldwide problem. It is less common in developed countries, and that is largely because of hygiene. One of the reasons that one of the areas where tuberculosis is very easily spread is in a household and in crowded areas like markets, crowded trains, this sort of thing. And that is more characteristic of developing countries. And so, unsurprisingly, tuberculosis remains very common in developing countries. About 10 million people a year develop tuberculosis, and of those, about 2 million will die. But astonishing figure is if you go and look at the worldwide population, you find 1/3 of the population have latent TB. That is, they have a consolidated tubercle, at least one, in their lungs that may remain latent for their whole lives and never break out. Eventually, if a tubercle remains that way long enough, it will eventually become invested with calcium phosphate and become calcified and then not be able to cause disease. So, but 1/3 of the human population will show the presence of latent infection, which is an astonishing figure. Now remember, in the latency, it is the activity of the immune system which is keeping it suppressed. It's keeping that tubercle single and small. Mess with the immune system, that tubercle can very easily break out and cause disseminated tuberculosis. This is one of the reasons why there is such a strong association with HIV, with human immunodeficiency virus. As an immune system collapses, so it can lead to this 1/3 of the human population becoming extremely vulnerable to tuberculosis. And this is the terrible thing that we see happening, especially in Africa, India, and parts of Asia. In south of the Sahara, many people have latent TB, but they also have extremely high HIV infection rates. In South Africa, more than 50% would test positive for HIV. And as people progress to AIDS, so the tuberculosis blooms and people become infectious. They eventually die, and they die of the tuberculosis itself, but not before they have spread it very widely. For this reason, accompanying the HIV epidemic, especially in Africa, has been an epidemic of tuberculosis as well. The two are very intimately linked. Okay, I'm going to stop there for the moment. I'll finish this and move on to our next section on Monday.